Assalamualaikum warahmatullahi wabarakatuh. Shalom Om Swastiastu, Namo Buddhaya, Salam Kebajikan, Rahayu. Let us praise and give our utmost gratitude to Allah Subhanahu wa Ta'ala for His mercy and blessings we could gather in this very special occasion. Good morning and welcome our speaker, Jesus Fernandez Soho, MD Ph.D., Director Cell Processing Laboratory and Barcelona Cord Blood Bank, Laboratory and Collection Director, Barcelona Cord Blood Bank, Blood Bank and Tissue, BST, Cell Therapy Unit, Spain. Dr. Nyoma Suci Widyastiti, Magister of Health, Clinical Pathologist, Consultant of Hematology and Immunology, PhD, the Head of Clinical Pathology Residency Education Program, Faculty of Medicine, Universitas Diponegoro, Semarang. Dr. Dwi Retnoingrum, Clinical Pathologist, Consultant of Infectious Diseases, the Head of Clinical Pathology Department, Faculty of Medicine, Universitas Diponegoro, Semarang. Dr. Maherdika, Clinical Pathologist, a teaching staff, Faculty of Medicine, Universitas Diponegoro, Semarang, as well as the moderator of today's guest lecture. Our respected professors, attending doctors and fellow residents, and all the audience, thank you for coming here today. Praise to God the Almighty, we have been graced by the chance to gather in this special occasion. A virtual guest lecture of Clinical Pathology, Medical Faculty, Universitas Diponegoro, Semarang, evolving stem cell labs to deliver advanced therapies. A stem cell laboratory is a specialized facility dedicated to the study, cultivation, and application of stem cells. The stem cells are manipulated to specialize into specific types of cells, such as heart muscle cells, blood cells, or nerve cells. The specialized cells can then be implanted into a person. Clinical Pathology, Medical Faculty, Universitas Diponegoro Semarang study program seeks to add the latest knowledge related to challenges in evolving stem cell labs to deliver advanced therapies by inviting a guest speaker, Jesus Fernandez Soho, MD, PhD. We hope that this event can provide new insight and knowledge in the utilization of evolving stem cell labs to deliver advanced therapies in the future. To begin with, let's start by sending our prayers. Prayer begins. Finished. Before we proceed to the main event, I would like to inform you about our agendas today. Opening speech from Dr. Nyoma Suci Widyastiti, Magister of Health, Clinical Pathologist, Consultant of Hematology and Immunology, PhD, Head of Clinical Pathology Residency Education Program, Universitas Diponegoro, Semarang. Lecture from Jesus Fernandez Soho, MD, PhD, about evolving stem cell labs to deliver advanced therapies, moderated by Dr. Maherdika, Clinical Pathologist. Discussion, photo session, and closing statements. The participants of this lecture will be given a certificate by filling the required data on the attendance link in the chat section. The first attendance link is now open. The second will be opened at the end of the discussion. The next agenda is opening speech from Dr. Nyoma Suci Widyastiti, Magister of Health, Clinical Pathologist, Consultant of Hematology and Immunology, PhD. To the Honorable Dr. Suci, the time is yours.
Thank you very much, Dr. Nita. Assalamualaikum warahmatullahi wabarakatuh. Good morning from Spain and good afternoon for all the audience in Indonesia. The Honorable Jesus Fernandez Soho, MD, PhD, Director of Cell Processing Laboratory and Barcelona Cord Blood Bank, Laboratory and Collection Director, Barcelona Cord Blood Bank, Blood Bank and Tissue Cell Therapy Unit, Spain. To the Honorable Dr. Dwi Retnoingrum, Clinical Pathologist Consultant, the Head of Clinical Pathology Department, Faculty of Medicine, Universitas Diponegoro, Semarang. The Honorable Dr. Mabru Ratusania Maherdika, clinical pathologist, staff of Clinical Pathology Residency Program, Faculty of Medicine, Universitas Diponegoro, our respected professor, attending doctors, resident, and all participants, thank you for joining us for this event today. Alhamdulillah, let's thanks to Allah Subhanahu wa Ta'ala, so that this day we could join here in this special occasion, a guest lecture of Clinical Pathology Residential Program, Faculty of Medicine, Universitas Diponegoro, Semarang, with the title of Evolving Stem Cell Labs to Deliver Advanced Therapies. On behalf of Clinical Pathology Residency Education Program, Faculty of Medicine, Universitas Diponegoro, I'm privileged and deeply honored to welcome our special guest, Jesus Fernandez Sojo, MD, Ph.D., Director of Cell Processing Laboratory and Barcelona Cord Blood Bank, Laboratory and Collection Director, Barcelona Cord Blood Bank and Tissue Cell Therapy Unit, Spain. We would like to express our heartfelt and highly appreciation of Jesus Fernandez Sojo, MD, Ph.D., for accepting our guest lecture invitation. We do hope you in the future can visit us in Indonesia, especially in Semarang City, in Karyadi Hospital and Clinical Residency Program in Faculty Medicine, Universitas Diponegoro, Semarang, Indonesia. This is the first international guest lecture in this year. We already have planned 3 guest lecture. The first one is this session from expert from Spain, and the second is from Netherlands, and the last one maybe from Malaysia. From this occasion, we intend to develop our student and quality and knowledge. We hope that the collaboration and networking between blood bank and tissue cells therapy unit Spain and Faculty of Medicine Universitas Diponegoro and Dr. Karyadi Hospital will continue. To all participants, thank you for joining this program and have an enjoyable and interesting guest lecture. Wassalamualaikum warahmatullahi wabarakatuh. Thank you very much for all participants.
Thanks to Dr. Nyoman Suciwidyasiti, Magister of Health, Clinical Pathologist, Consultant of Hematology and Immunology Ph.D. To all participants, please fill in the attendance link which can be accessed in the meeting chat. Ladies and gentlemen, the next agenda will be led by Dr. Maherdika, Clinical Pathologist. Allow me to read her curriculum vitae. Dr. Maherdika. Her educational history is a general practitioner at the Faculty of Medicine, 11 Maret University, Surakarta, and clinical pathologist at Faculty of Medicine, Diponegoro University, Semarang. Her work experiences are clinical pathology specialist doctor at Karyadi Hospital, Semarang, hematopoietic stem cell team, Karyadi Hospital, and clinical pathology teaching staff, Diponegoro University, Semarang. To the Honorable Dr. Maherdika, the time is yours.
Thank you, Dr. Nita. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, our professors, doctors, seniors, and residents. Welcome to our annual guest lecture series, and I will be serving as moderator today. Today has joined with us our speaker from Barcelona. Please welcome Dr. Jesus Fernandez Sojo, Ph.D. Good morning, Dr. Jesus.
Good morning,
Yeah.
Ika. Thank you very much.
We are very grateful to have you join us here. Thank you very much for your time. And now, before you start the presentation, please allow me to read your curriculum vitae. Jesus Fernandez Sojo, MD, Ph.D., was graduated in 2010 for a degree in medicine from University of Barcelona, Campus Belviche. In 2015, graduate for specialist in hematology from University of Hospital German Chef Ipuzol. And in 2023, he graduated for doctoral thesis in medicine from University of Barcelona. And for work experiences, from May 2011 until May 2015, he was resident in University Hospital Germansgraaf Ituyo, Institut Català de la Salut, and until April 2019, he was Medical Director Donation and Apheresis Collection Blood Bank and Tissue, University Hospital Vall d'Hebron, and until April 2024, he was Medical Director of Cell Processing Laboratory Medical Staff in Ph.D. And now he is a Director Cell Processing Laboratory in Barcelona Cord Blood Bank, Laboratory and Collection Director Barcelona Cord Blood Bank, BSD Cell Therapy Unit. Now, moving along to our main session, today we will hear a lecture by Dr. Jesus about evolving stem cell lab to deliver advanced therapies. And after the lecture, we will have a discussion session. Please, Jesus, the time is yours.
Thank you very much, Dr. Dika, for inviting me to participate in your clinical program of pathology study program, Universidad de Goro. And thank you, Dr. Nyoman Suchi, for your kindly presentation. I'm going to try to share my presentation. Okay, I think you all see my presentation.
Yes, we have. Thank you.
Okay, so yeah, thank you again. It's a pleasure for me to be here. And the topic I'm going to talk about is the evolution of stem cell laboratory to deliver advanced therapy. So as you mentioned, I'm recently the director of cell therapy service at the Banc de Sang i Teixit of Barcelona. This is a little bit of the overview of my presentation. So I will do an introduction, conventional cell therapy laboratory and their missions, and what are the similar things regarding advanced manufactured products. And then I will focus for those steps that are similar in stem cell transplantation and in advanced therapy medicinal product like cell collection and cell processing, the automation equipment that can be shared between both laboratory, the quality control of cells and medicinal advanced products, how is the supply and administration, a little bit of regulation and good manufacturer practice, some recommendation or suggestion of how to develop a cell therapy program. I will show you a little bit of our experience and our model. Finally, some slides about how to develop a laboratory to manufacture advanced therapy. And finally, I will conclude with some key of the topic. So to start with a little introduction, as you all know, the stem cell transplantation started in the last century, and the main mission of cell therapy laboratories to provide stem cells for transplant, and laboratory process those stem cells and adjust them to be administered to the patient. So sometimes it is needed to plasma reduce the product or to deplete red blood cells, and in autologous setting to cryopreserve the cells. This is the basically process that cell laboratory does. Later, there started to appear some equipment to allow the cell positive or cell negative selection of certain types of cells like CD34 to boost or to deplete T cells like CD45-Ra or alpha beta in order to mitigate some complication of transplant like graft versus host disease. And later, in the 90s decades, started to appear the TSF to mobilize peripheral blood stem cells and to collect from blood, peripheral blood, and started also the DLI, donor lymphocyte infusions, to treat relapse and to treat the immune recovery when the patient is a mixed hematism. And then started the umbilical cord blood banks. So in this slide, you can see the typical flow chart in stem cell transplantation. So at the beginning, we have a patient who is mobilized with GSF or with chemotherapy. And then there is a release of stem cells from bone marrow into bloodstream. Those cells are connected to a medical device, a Persis device. You can see in the slide that regarding the size and the density. This procedure takes between 4 to 6 hours, and the device is available to collect the stem cells, and then those stem cells has to be processed before infusion to the patient. In autologous setting, the cells are plasma reduced, then is prepared the cryomedia, and is cryopreserved the cells and storage in a liquid nitrogen tank or vapor phase tank until the patient is ready to receive the cells. In that moment, the cells are removed from the storage tank and then is thawing into a dry device or water bath, whatever, and has to be thawed quickly and then has to be infused into the patient in less than 1 hour. So in the last slides, we saw the conventional stem cells transplant, but recently processing laboratory started to manufacture other cell types. You can see here in this slide, modified cells, natural killer cells, macrophage, T cells, tumor infiltrative lymphocytes, and those cells are manipulated with expansion or engineering modify genetically the expression of the receptor. So this is considered a major manipulation, and then this is considered a drug. And what we see in the next slide, the regulation is more restricted. And moreover, these advanced cell therapy, they have some challenges. Some of them is the cost associated with the production can be really high. Sometimes the resources are limited, basically in academic hospitals. And this treatment has to be manufactured following the good manufacturer practice regulation that is more restricted than the T cells, the tissue and cells regulation. But what about when external manufacturer started to produce CAR T cells? Basically, here in this slide, this is an example of this cell from Novartis. This is Kymriah. And in this slide, you can see on the left, clinical steps that it is expected to be done by the hospital. And on the right, the manufacturing steps that usually is done by industry. So the method used for collecting and processing stem cells can be adapted for CAR T cells, are really similar. We will see them, basically, the leukapheresis and cryopreservation. So the hospitals may be able to provide this starting material for the industry, and sometimes the manufacturing facility could be the hospital, but this is not always like this because the high cost and the complex of the procedures. So what about the sources of stem cells? There are basically three sources: apheresis, bone marrow, and cord blood. We will focus in apheresis source for the production of advanced cell therapy. So if we focus in this step, we can see that the centrifugation of the cells achieve the collection of lymphocytes and monocytes that the size and the density is similar than peripheral blood stem cells. So the procedure can be the same. In contrast to transplants, the main source is T cells in order to manufacture the CAR-Ts. And what is the factor that impact in the amount of T cells? Basically, the patient recount lymphocytes T cells in peripheral blood, and also the amount of total whole blood that goes through the device. Here in this slide, you can see the flow chart for the starting material to produce an advanced cell therapy. So the first step, as we have said before, this is the collection. Collection for obtaining T cells takes more or less between 4 and 5 hours to achieve the amount of cells predefined by the manufacturer. Once the product is collected, then the next step is the labeling. This is really important to maintain the chain of identity. So most of blood banks or cell therapy laboratories use an international society. Is called ICBT 128. This is an international society of blood transfusions for the labeling. So every collection has to assign a donation identification number, and the label has to be a specific format, and all the product has a code that we can understand simply with this code what is the product. So this is the way how blood banks can maintain the traceability and then all of the transformation. So the next step depends of the manufacturer. Sometimes the manufacturer want fresh product. So in that case, the courier pick up the leukapheresis, and then this product is shipped to the manufacturing facility to produce the CAR-T. In other cases, the manufacturer wants the centers to cryopreserve the product. This cryopreservation is very similar than the cryopreservation of stem cells. So the first step before cryopreservation used to be an adjusted of the volume. So plasma reduction is recommended to reduce the volume and also to increase the white blood cell concentration, because usually these leukocytopheresis, the white blood cells are low because the donor is not mobilized. So in our case, we use an automated device, but a centrifugate can be used. Then the cryopreservation solution is prepared with cryobags and then is packaged in a cassette. After that, the product has to be cryopreserved. The device that usually use cell processing laboratory works with nitrogen vapor phase. And then once the product is freezed, then has to be storage in a vapor phase tank that is control the temperature continuously. And then we wait until the manufacturer or the sponsors schedule the pickup. Then they provide a dry shipper. We remove the cryo bags from the nitrogen and we pack it in that dry shipper. Then is shipped by road until the cell manufacturer facility. Here in this slide, you can see our history of collection stem cells for starting material for CAR T cells. So in gray color, you can see the collection for clinical trials. In orange color is collection for Yescarta from the manufacturer Kite Gilead. And in blue color is the collection for Kymriah from Novartis. So in 2016, one of our centers participated in Eliana clinical trial from Novartis that makes possible the approval in the FDA of Kymriah years later. So in Spain in 2018 was approved Kymriah for commercial setting, and in 2019 was approved Yescarta for large cell lymphoma. And then, as you can see, the activity has maintained or growing up a little bit. We have performed more or less more than 500 collections. So most of them are from clinical trial, but basically the most collection are for Yescarta that seems to have better results than Kymriah. And how we manage to incorporate this activity with our researches. So in this slide, this is a paper that our group published in Cytotherapy Journal explaining our model. We call it hub and spoke. And as you can see, there is a centralized cell processing laboratory, single laboratory, and medicine warehouse. So in this central hub, we receive the leukapheresis that has to be cryopreserved from different collection centers that belongs to our institution. And also we receive the final product manufactured by the industry. We store it in our hub, and then we ship it to our spokes. In the dashed line, this is our institution, our tissue establishment, Catalonia Blood and Tissue Bank. So the hub and collection centers belongs to the same institution. Collection centers, as you can see, they are inside the hospital, but they belongs to our institution. So what we do is we make agreements with hospital because they are another institution, and we define the responsibilities of each part. So they make us the possibility to incorporate these CAR T cells like Kymriah, Yescarta with a really fast qualification because the standard operation procedures, the quality system is the same in all of the collection centers and in the hub, in the central facility. So in some cases, cryopreservation is required. And what are the advantages of cryopreservation? It permits flexibility. So what does it mean? It means that some cases, for instance, patients who would need a Kymriah in a few weeks or in a few months, but at this moment, it doesn't meet the criteria. The collection can be done in this moment that maybe fits better, and then be cryopreserved until the patient has the criteria to be manufactured the product. So we use cryoprotectants. We use DMCO at 10%. This is to prevent damage of the cells after cryopreservation for different mechanisms. We use an equipment, a control rate freezers. In this slide, you can see the curve of the cryopreservation that has to be done between 1 to 5 degrees per minute. So this is really important to maintain a constant cryopreservation to achieve the maximum recovery of cells in cryovials. And after cryopreservation, cells typically are stored in a single or more cryobags in a liquid nitrogen or in vapor phase nitrogen liquid. We used to prefer for advanced products to use vapor phase nitrogen liquid because manufacturers doesn't put an overwrap bag, so they used to send the cells with only the primary bag. So if liquid nitrogen is used, maybe can produce cross-contamination. So we use vapor phase in that situation. And what about the equipment? As you all know, the cell therapy processing involves so many steps, so many procedures like plasma depletion, cell separation, washing in some cases, or major manipulation like culture, transduction, or expansion. Those procedures can be done with a manual system, but in that cases, a high variability would appear even with intensive staff training. So what are the advantages of using automated instruments? It can reduce the variability and reduce the risk of microbiological contamination, reduce the number of staff, but in contrast, increase the cost of the equipment. The equipment is expensive. Also, the suppliers and the reagents. In the picture, you can see different mechanisms of technology used to centrifugate cells and to wash the cells. Basically, the equipment works with a centrifugation system, very similar than collections devices. In some cases, the system is by elutriation, so there is a chamber that concentrates the cells and avoid to lose other cells like platelets. And in other cases, the device uses ultrasonic acoustic waves or by filtration. But this is not very common, but it's possible. And one step, really important step, is the quality control. So here in this slide, you can see the quality control that is used to perform for the starting material to produce advanced therapy. So this is for the leukapheresis. We test cell blood count to count the number of white blood cells, hematocrit, red blood cells, and platelets. Normally, the manufacturers require a minimum of white blood cell count in leukapheresis or a minimum of mononuclear cells or T cells. So it's important to test the identity and the purity of CD45, CD3, in some cases CD4 and CD8 by flow cytometry. We also test the viability with traditional method, with tryptan blue, that works with dye exclusion. Or recently, we introduced in last years an intracellular method to test the viability with 7-actinomycin D. And finally, it's important all the steps that you manipulate the cells, you have to test the sterility. We test back alert hemocultures to identify bacteria and fungus. And what about the quality control of advanced therapy medicinal product? There are more tests that should be done. Highlighted in the red box, you can see those tests that are shared between conventional laboratory and advanced therapy medicinal product laboratory. So sterility is the same with back alert, and the identification and the purity of cells are performed with flow cytometry, so it's the same, but there are other tests, additional tests, more complex that should be done, but what I mean is the same quality control would be able to do some, at least, test and to be prepared to do more tests for advanced therapy. Once the product is manufactured, is shipped back to the site. So the manufactured product is cryopreserved and then is shipped in a dry shipper. Again, is scheduled with the clinical site, is received the product and then transferred to the final location in the vapor phase tank until the day of the infusion. The infusion of advanced therapy product is very similar than blood transfusion. So it's to be done quickly and is used to be give prophylaxis with antihistaminic and antipyretic, and the adverse reactions, some of them are similar than blood transfusion, for instance, or stem cells, the adverse reaction from DMCO or cellular debris, but in that cases, other reactions are secondary to the therapeutic cells, cytokine release syndrome or reversible neurotoxicity. And a really important point for cells and advanced therapy is the regulation. The regulation of the advanced therapy medicinal products, such as CAR T cells, is different than the regulation of stem cells. Advanced therapy products are regulated like drugs. So medicinal agency has to approve them, and laboratories who manufacture this kind of treatment should be authorized by these agencies. And they have to follow the good manufacturer practice that are more stringent regulation than T cells. So the first steps before manufacturers are regulated under the tissue and cell regulation, tissue establishment, at least in Europe. And they have to follow international standards like JC. And recently in Europe was approved the Sojo regulation substance of human origin that has to be met in the next 3 years. So in that regulation, refer to the European Directive Quality Manager technical requirements to be followed for the manipulation, the collection, the quality control, the traceability. So refer to this directive and will be the same for all the European members. So this is a really good news for all European members to follow the same directive, because what happened right now? Right now, what happened is we have so many directives for the starting material, for the collection and the processing and the cryopreservation of the raw material. And those regulation applies to the collection, testing, processing, storage, distribution, the safety, the adverse events or adverse reaction. This is biovigilance, and the traceability. So what about is if some centers want to develop a cell therapy program or wants to start? The first step is to develop a stem cell transplantation program and to create a cell processing laboratory associated to this transplant program. Our suggestion is to consider to affiliate this cell processing laboratory to the blood collection and processing centers. And why? Because blood bank staff knowledge and skills and blood bank laboratory equipment can be shared between blood bank and cell processing. So the collection device, the donor screening for infection markers disease are really similar. So this is a way to save resources. And then the centers has to define a product quality standards. And those can be similar than in blood. What are the quality standards that one can expect to find in a centers? The cross-contamination ratio, the product recovery, the white blood cells recovery after processing, the storage, the labeling, the distribution, and there is a guide in Europe that is really complete and provide all of this information, that this is the European Directorate for the Quality of Medicine and Health. There is one for blood, and this is another for tissue and cells and advanced therapy. So this is really interesting. And I'm going to talk about a little bit about our model. Here we are a public blood bank that provides service to 7.5 million inhabitants in Catalonia. And there is only one blood bank, and their mission is to guarantee the supply and the proper use of substance of human origin. And we have a headquarters here. You can see in the center of the slide that this is the place where we receive all the whole blood collected. This is processed here and supplied to the hospitals. The hospital, they have a local blood banks that also belongs to our institution. They have a little inventory blood components, and every day or periodically they check the inventory and ask for more components if they need it. And also in the headquarters we have the main laboratories, immunohematology to identify difficult antibody, irregular antibody, HLA laboratory, infection disease laboratory, and also the cell processing laboratory is in the headquarters. So our institution, what did when create the cell processing program, they follow the same model. So to centralize also the cell therapy program. This is not typical, even in Spain or in Europe. The most typical in the rest of Spain and in Europe is that each hospital has its own cell processing laboratory. But in our case, we focus, or we manage to centralize also the cell therapy program. So this has some advantages. This is also has some challenges, but this is how we work. So in this slide, you can see highlight in a red box, the stem cell laboratory is in Barcelona capital. And then this laboratory provides service to 11 transplant centers. One of them, some of them, they have only autologous stem cell transplantation program. So they are small and in the peripheral of the territory. We provide also service to 3 pediatric hospitals, and also we provide service to adult hospitals. It has ICO hospitals, by the Bron, Hospital San Pau, with autologous and allogenic transplant program. So it make us possible to perform more than, or to provide more than 500 transplants per year. So this make us possible to invest in equipment in order to be able to process a high number of products. And what happened when CAR T cells arrive in Spain? So what happened is that the manufacturer has to qualify centers to be able to use their CAR T cells. So in Catalonia, they were selected the centers with allogenic program to treat patients with CAR T cells. So the hospitals where they are located in the Barcelona region, centralized also. 6 clinical sites, 4 with adults and 2 for patients. And we were the facility to process the starting material if it is needed for Kymriah, and to centralize the warehouse medicine. What does it mean? It means it in Spain and in Europe, CAR T cells are considered a drug, and then has to be received by a pharmacy. And our facility is not a pharmacy. So what we did is we did an agreement between the pharmacies of the hospitals in order to do the reception and the storage of the final product on behalf of the pharmacies. So we receive the products, and then we provide all the documentation, the pictures of the cryo bags, and we expect that the pharmacy of the hospitals approve this reception. So this is what we do. And this is the map of the centers that are authorized to treat patients with CAR Ts in Spain. So as you can see, there are more than 20 hospitals, but most of them are in Catalonia. So, and at the beginning, most of the hospitals were authorized, but they were not qualified by the sponsors. So in Catalonia, yes, we started really fast to increase our activity. And what were the criteria to authorize one centers or others? Well, you can see here the criteria that the health minister in Spain used to decide what centers could be able to treat patients. So they required to have a multidisciplinary unit with all the professionals involved in the process. Also, they required the activity of allogenic transplant with complex procedures. That it means unrelated donors, umbilical, cord blood, or haploidentical donors. They require also to have the JC accreditation, a multidisciplinary clinical pathological committee, to have a certificate of good manufacturer practice. So our institution we will see later, we have the good manufacturer practice to manufacture some drugs, no CAR-Ts at this moment. So this help us to clinical sites to apply to be selected. Also the activity of apheresis stem cells, the total activity of complex cell processing, that it means CD34 selection or CD45 RAD depletion. So this is something that our laboratory also perform and has a preclinical experience with immune effector cells. And then were selected. And this is some pictures of our facility. This was when was set up. So this is the classified area. It has a 200 meters, more or less. And this is the room that we use to process. These pictures are empty, but we'll see later with the equipment. This is a map of our classified area that we use. Cell processing laboratory, our manufacturer laboratory, they have also specific rooms, and tissue bank. We have also important tissue bank, and the different colors is the classification of the room. So in Spain, it is possible to process in a class D. So this is where we process cells even for advances therapy medicinal products. So the plasma reduction and the cryopreservation of the cells before manufacturing is performed in a class D. So this is allowed in Spain, but the manufacturing of our drugs, we manufactured virus-specified T cells, mesenchymal stem cells. They have to be performed in a class B or C. So this is the difference. And the difference in the class is regarding the number of particles are in the environment and the size of these particles, viable particles and not viable particles. And this is a picture of the room that we use to process stem cells for transplant and the starting material. So it's a class D, it has 40 meters more or less. We have 2 biological safety cabinets, and we have all the equipment to facilitate the processing. In our case, we use this device. This is CEPAX from Cytiva. This is a flexible device that it has different program, cell plasma depletion, Bafiquat to process bone marrow, washing, and others. And also we have other equipment for the addition of the cryopreservation solution to control the rate of the addition of the cryo media and to control the temperature and to homogenize. And finally, up you can see the control rate freezers. We use it nitrogen control rate freezers that control the rate of freezing. And this is the equipment that we have for both, for standard cell processing and for advanced manufacturing. So we have centrifuge just for backup, Clinimax Plus for cell selection for CD34, Clinimax Prodigy for the selection and expansion, and G-Rex for expansion of T cells and other bioreactors. And this is the map of our cryogenic area. We have 11 nitrogen tanks for stem cells that we have storage in the last years. So these tanks, it has a capacity of 400 of bags, more or less. Sometimes this is very difficult to achieve that physicians in charge of the patient give us the consent to remove the bags, but we need it because the space is not unlimited. And on the right, you can see this is a vapor phase nitrogen tank specifically dedicated for advanced product. So we only storage here the starting material cryopreserved that is not shipped to manufacturer. In some cases, for Kimbraya, we collect cells, but the manufacturer has a upper limit, so they recommend not to provide more cells that they needed because it doesn't improve the manufacturing and they consider excellent material. So in that cases, we don't ship it, we store it. And these cryo bags could be used again during the next 18 months. So if the patient, something happens or the patient needs a second manufacturing, it is possible to send this cryo bag, but Novartis only has validated until 18 months. And here we have not only CAR T cells, TCR, mesenchymals, stem cells, virus T cells specified. So something that has to be taken into account is that the manufacturer provide the final product with different containers. Sometimes, or most of the cases, are in cryo bags, but sometimes are cryovials. So you have to be able to have different racks. So sometimes this is complex or challenging because you need the space for the specific racks. And this is control the temperature continuously. And if any excursion temperature alarm appears, then we have security in the building and they call the person on duty to advise that there is an alarm. And as our cell processing center is not close to the clinical site, sometimes there is 20 kilometers or even more, 300 kilometers. So we need to use a cryo shippers. So this is an example, the cryo shipper, and how does it work that maintains the cryogenic temperature under 150 degrees during 10 days, more or less, and with a temperature sensor that the center checks the display in order to make sure that there were no alarm excursion. And this is the equipment for the quality control. So we have hematological blood analyzers, cytometries, cultures, and we have other laboratory supports like HLA for histocompatibility, immunohematology, and microbiology to test the sterility. And this is the international accreditation. So regarding cord blood, there is a specific standards. This is called FACNECORD. It's an American standards, really restricted. And in Spain, we have another standards for cord blood. This is called CAT. We also have for stem cells and immune effector cells, JC standard. This is an international, at the beginning, it was European, but right now they make an agreement with FAC, and now it's an international standards for transplant programs, but also for those of them who wants to apply for immune effector cells like CAR T. So this is a good way also to develop a transplant and a CAR T program. We receive constantly qualification for manufacturers. So this is really burden to be continuously qualified. So there are some projects like GoCart that they mean to reduce the necessity of to be qualified for each manufacturer because some of them require some different things. some different target cells. They have a specific platform, so this is really time consuming. And for the manufacturer in our facility, we have to follow the good manufacturer practice and to be authorized by the Spanish Drug Agency. And I would like to talk about the cord blood bank. So in Barcelona, we have one of the most important cord blood banks in Spain, but also in Europe, thanks to my last boss, Dr. Sergi Querol. So our Barcelona cord blood bank, we have right now more than 20,000 of cord blood units cryopreserved and ready to be used for any patient around the world that would need a transplant with a cord blood unit. So we provide this information to the Spanish bone marrow registry, REDMO. And from all of our history until this year, we have provided more than 2,000 cord blood units for transplant. So this is more or less the rate for transplant. So you need a high number of units in order to ship units for transplant. And this is the map of Catalonia where we receive, or from where we receive cord blood units. So we not only receive cord blood units from Catalonia, where this is where we stay, but also from Andorra country, Balear Island, and other communities like Aragon, Navarra, Cantabria, and Extremadura. We qualify their maternities and we made an agreement with all of them and to be able to increase our activity. So in this slide, you can see the activity of our cord blood bank. Our cord blood bank was created in 1995, and the activity increased, but not too much until 2007. In this moment, we create the Concordia program. Concordia program is what we saw in this slide. We make a collaboration with other communities in Spain in order that we receive more cord blood units. So in that time, increase the activity of cord blood, and also during this time, the activity of cord blood transplantation was very high. So this is the reason why we receive a lot of cord blood units, because we ship a lot of cord blood units. So after 2013, the activity started to decrease. We increased the criteria to process cord blood for transplantation, and the reason of this decrease was basically the status of transplantation make possible to patients that they didn't have donors to have sisters or fathers or children to be at least transplant. So and other reasons make that the activity of cord blood bank decrease, and we develop another concept of cord blood bank we call 2.0 in order to take profits of those units that doesn't meet the white blood cell criteria, to use it for dry ops or platelet gels for patients with ulcerous skins or other application, red blood cells for transfusion in preterm newborns. But the new futures that presents to cord blood transplantation, we can see in this slide. So when we receive cord blood, we receive the bag, but we also receive the umbilical cord, the Wharton jelly. So from this Wharton jelly, following good manufacturing practice, we can develop mesenchymal stromal cells. And the application of these mesenchymal stromal cells is for regenerative medicine. We use them to treat patients with graft versus host disease in a compassionate usage. And we also use it for cardiac disease. So there is a quite potential of these mesenchymal stromal cells. But other application of cord blood units are another advanced therapy. For instance, we have a project that we obtain induced pluripotential stem cells from cord blood unit. So these kind of cells can be reprogrammed to further generate specific cells. So from iPS, you can develop NK cells, T cells, or tissue-specific T cells. So as we are going to see in the next slide, but we have a master bank of homozygous HLA iPS. What it means is in our cord blood bank, we know the HLA of the units. So we make a study of the HLA most common in Spain, and we select those units homozygote for the most typical HLA in Spain. So in order to, in the future, can treat patients and respect the HLA in the 70 or 80% of all the patients. Another project that we are starting to receive from our college in the hospital clinic is the development of CAR NK. So we update the consent inform for the cord blood donors in order to incorporate the possibility to provide products to develop drugs and to treat patients. So in that case, can be a CAR NK for anti-tumor effects, but we are collaborated with a hospital clinic. They are developed an NK cells for antifungal treatments. And finally, also cord blood cells, they have abundance of immune cells, naive cells that can be used off the shelf and to develop T cells to treat infection after transplantation, or to modify genetically to develop a CAR T also for cord blood unit. So this is an open door that we present for in the future. And this is an example of our project. We call it IPSPANIA. It means induced pluripotential stem cells. And we have haplolines. So we collaborate with researchers that they develop a CAR T, and then we are able to these induced pluripotential cells reprogram to develop NK, and then to transduce the CAR T. This is now the master bank that we are developed. Then we will start a preclinical validation, and finally, if everything goes well, we will manufacture the working cell bank in order to be applicable to patients. And those are the projects that we are working to develop a universal CAR 1920 NK to treat leukemia or B lymphoma, another CAR T for solid tumor to treat tumors that express HER2 protein, and also developing antifungal CAR Ts. So this is a summary of the advantage of our model, of a centralized model. So the basically advantage is the harmonization of the apheresis. So in all of our collection centers, they use the same SOPs, the device are the same, the training of the staff is the same, and the quality control laboratory that test the collection are the same. So it allows us for a rapid qualification when manufacturer starts here in Catalonia. The results are centralized, so this is really helpful for the health care. So we have one unique biostorage area with 2 dedicated nitrogen tanks for advanced therapy. This is the number of lab technicians that we have, 8 lab technicians, 4 in the morning and 4 in the afternoon, because we process in the morning, in the afternoon, 2 qualified persons to release the product, 2 biotechnology consultants, 2 hematologists, and 1 technical director for the warehouse medicine. We collaborate with a large number of different sponsors, so we are used to receive different products. The potential challenge is the infrastructure, because the activity is increasing, so we have a capacity, we are increasing, but maybe there will be a moment that we will have to invest. And this model opened some opportunities, like this one, that there was 1 collection centers in Spain, 500 kilometers far away from our institution, that they were selected to treat patients with Kymriah, but their cell processing facility was building, so they couldn't process. So we create a collaboration with them, and they collect the cells in their collection centers and ship those cells to our facility to be cryopreserved, and then we provide to Novartis. So it was quite new in Spain and in Europe. But are all this model possible? The answer is no. This is a paper who makes a survey on hospital practice to support external manufacturer products like Kymriah or Yescarta. What was the model? So the service was done in USA, basically in Canada, Australia, in France. And as you can see in the table, the most typical model to support this externally manufactured cell gene therapy product was the stem cell processing laboratory that also provides service to transplant programs. This was the 78%, so most of them, this is the model. Another model is that the advanced therapy laboratory of the clinical site also receive the external manufacturer product. This is another possibility, or the combination of the advanced therapy or cell therapy program, or to create a specific program for this proposal. But again, I think this is important to share staff, to share resources in order to make possible to incorporate this very expensive therapy. So regarding establishing a laboratory to manufacture an advanced product, as you all know, to processing graft has to be done in a biosafety cabinet and in a controlled environment, less restricted for stem cells. But if we want to create a manufacturing facility for advanced product, then the area has to be in a high standards. So the classified area can be shared, but as the regulation is different, for advanced therapy is more restricted. And this is expensive to build and to maintain a good manufacturer product facility. So something that can be done to reduce the high requirements is to use automated systems in order to make all the process in a closed system and be able to produce the product in a class C, for instance, that is not too restricted than class B. And finally, this is a paper with the cost of the steps to process product for transplant or for CAR-T. So as you can see, the steps that are not represented a major manipulation like leukapheresis, cell separation, washing, plasma depletion, or cryopreservation. They are expensive, but not too much in compare with all the procedures like enrichment, activation, viral vector transduction, expansion. This is really expensive. The equipment, but also the patient cost. So yeah, this is a combination that you can have to only do the procedures that are not a manufacturing procedures. And this is the advanced products that our laboratory provide to clinical sites. So on the right, you can see viral T cell. This is a virus-specified T cell against cytomegalovirus, Epstein-Barr virus, herpes human virus, BK virus. So in colors, there is the activity of each year. So this is something we are increasing years. So this is viral infection. At the beginning, we use it for transplant patient, but we are now using even for patients with immunodeficiency disease or other non-transplant disease. We also provide mesenchymal stem cells to treat graft-versus-host disease or immune cytopenias or regenerative usage. We use cells CD153 for infertility disease. The clinical trial called eGenomic, Pericord for mesenchymal stem cells for necrotic heart disease. And we are also developing a TIL, tumor infiltrative lymphocyte for tumor solid patients. So we treat patients. So this is the beginning. And finally, the conclusion is, as you all know, the field of cell therapies has increasing a lot over the 2 decades. At this moment, we have transplant and advanced therapy product. So transplant procedure was the only one in the last 50 years, and it remains unimportant. Some discussions appeared when started CAR-T cell that transplant was going to disappear, and we didn't see that. Even we increased the activity for the transplant. So this is something that should be taken in mind, that the activity even is increasing, even with advanced therapy. And cell processing facility can be set up from hospital blood bank, sharing equipment, facility, and staff. CAR-Ts are increasing, and is a really good treatment for many hematological patients and non-hematological patients, and can be developing in the cell processing facility. And when external CAR-T cells are receiving in a hospital, the hospital has to establish the standard operational procedures for the labeling, transport, storage, product release, and cryopreservation. Something really important is from a regulatory point of view, it is different, the regulation of cells and tissue from starting material than those from the manufactured advanced products. And also take in mind the burden or the high activity to be qualified constantly by the manufacturers. And yeah, thank you very much. This is a picture of our staff and all of the physician in charge of the transplant program and advanced therapy program in Catalonia. And thank you very much for your attention. And let me know if you have any question that we can discuss. Thank you very much.
Thank you, Dr. Jesus, for the interesting lecture. And now, moving on to our discussion session. If there are participants have some questions, please don't be hesitant to write down on the chat column below or raise hand if you want to ask Dr. Jesus directly. Untuk peserta, apabila ada yang ingin bertanya dalam bahasa Indonesia, kami dari panitia akan membantu untuk menerjemahkan. Baik. Okay. First question from Dr. Priska. Good morning, Dr. Jesus. Can bone marrow transplant be done in thalassemia patients using those methods? Thank you, Doctor.
Yeah, thank you very much for your question. Thalassemia patient, this is an indication for bone marrow transplantation. So normally the transplant is done in a pediatric age. So the source usually is bone marrow. This is the most typical source for these patients. And right now we are being qualified from a genetic treatment to treat patients with thalassemia that considers in those patients mobilize and perform leukapheresis. These leukapheresis from mobilized stem cells are shipped to a manufacturer facility that is in Europe, outside of Spain, and they modify the stem cells in order to increase the normal hemoglobin, and then the product is shipped back to the facility, and the patient receives an autologous genetically modified transplant, and the results seems to be really good. So this is something to have present, that thalassemia is an indication for classical allogenic bone marrow transplant, but again, this is also an indication for genetically modified T cells, stem cells, sorry.
Okay, thank you. I want to ask more about the thalassemia. So is it possible to use the autologous donor you said before? How about the allogenic donor?
Yeah, in our setting, yeah, I don't have experience in autologous transplant, because probably, yeah, the disease, it doesn't disappear. We don't have experience with autologous. We only perform allogenic transplant in thalassemia. I think in the past, maybe a physician in charge require us to do a backup autologous, just in case it was a graft rejection or graft failure. But as a treatment in the last decades, we use it allogenic transplant here.
Oh, okay. Thank you. And second question from Dr. Sita. Good morning, Dr. Jesus. How effective is the use of HHC processing cell laboratory in stem cells? Thank you, Doctor.
Yeah, thank you, Dr. Sita. In stem cells laboratory is really important to guarantee the potency of the cells. This is our main objective. So when we process cells, we have to guarantee to recover almost all of the cells and to maintain the viability. In some cases, it's really challenging, because For instance, when we receive a bone marrow source and the patient has major incompatibility, this is really challenging because we have to reduce the number of red blood cells, but maintain the number of white blood cells because this is impact in the outcomes of the patient. So we have to adjust the device in order to maintain the safety of the patient and to prevent the hemolytic ABO disease by the number of white blood cells. And this is for bone marrow. For other procedures like washing or cryopreservation, it's really important that in that procedures, always you are going to lose some cells. This is impossible to recover all the cells, but you have to validate your procedures in order to increase the number of recovery of viable stem cells, because this has an impact in the transplant. So this is the most focus that the cell processing has to be in mind to maintain the potency and the viability, the sterility. So this is how we can effectively use stem cells from the laboratory point of view.
Thank you for your answer. Okay, and then the question from Dr. Tara. Is it possible for the body to reject stem cell therapy after all the screening procedures? Do patients with allogeneic stem cell therapy also receive immunosuppressive agents like patients who receive organ transplant? Thank you.
Yeah, thank you, Dr. Tara, for your question. This is possible even in autologous transplant program. A patient can reject the cells. This is not very typical, and we don't see in the last years. When this is most typical is in allogenic transplant, because allogenic transplant, even if you match the patient for the HLA, we match for 5 or 6 locus, HLA A, B, C, DR, DQ as a minimum. So you have to match the patient. Right now, starting to appear haplotransplant that match only the half, but there is always a risk in allogenic transplant to reject stem cells, even if there is a full match. Probably there are some more immunological reasons to reject the cells, and how you can prevent this rejection. You can prevent with matching HLA. Also, you can prevent with the conditioning that the patient receive influence in the risk of graft rejection. If you increase the conditioning, so if you use a myeloablative conditioning, the risk is lower than if you use a non-intensive conditioning. But in contrast, the more conditioning or intensive conditioning you use, the more toxicity will have the patient. So this is something you take into account. And regarding the second question, the immunosuppressive agents, allogenic patients used to receive after transplantation immunosuppressive agents for the prophylaxis for a complication that is called graft versus host disease. That this consists in the fact that the graft consider the recipient as an allogenic and then damage the cells. So this is a really mortal potency complication. So patients receive an immunosuppressive agents. In some cases, if you manipulate the graft ex vivo, I mean removing the T cells that are in charge of this complication, then you don't have to use it immunosuppressive agents if you manipulate the graft. But they have another complication that the reconstitution is low, but the immunosuppressive agents that the patient receive after transplantation is to avoid this kind of complication.
Okay, thank you. So for the advanced therapies with the ex vivo infusion, the immunosuppressive agent is not necessary, you think?
Yeah, in Europe, the regulation of tissue, cell and tissue are more or less the same in European countries with some difference because European members can adopt some difference, but right now with a new Sojo regulation will be the same for all the state members. And about your question, how is different? For instance, in Spain, to process stem cells or starting material for CAR T cells, Class D is enough. In Germany, I think they require to have a Class C at least. So there are some difference that probably with the new regulation will be the same for all European country. And regarding these two drugs, Kymriah and Yescarta, the question is how do they differ each other from the manufacturing procedures? Yescarta is performed in fresh. So that it means that the starting material is collected in the clinical site in fresh, no cryopreserved, and then is shipped to a centralized facility who perform an isolation of CD4 and CD8, and then is performed the modification. Kymriah, in contrast, Novartis, require always to cryopreserve the starting material. This is the basically difference. And then the other difference is regarding the manipulation that they perform to the cells. I don't specifically know what is the difference, but the clinical results is that Yescarta showed a better clinical outcomes than Kymriah. In contrast, Kymriah has low cytokine syndrome release. So our physician in charge, they used to administrate Kymriah if the patient is elderly or it's not so fit to receive a CAR T cells, but it seems that the results are better with Yescarta. This is what it seems, and they are approved by health minister in Spain and in Europe. So this is provided by the health care of Spain.
Thank you, Dr. Jesus. And that's for the Kymriah and Yescarta. And Dr. Marcel, do you want to ask more to Dr. Jesus? You can do it directly to Dr. Jesus.
Okay. Thank you, Dr. Dika. Good morning, Dr. Jesus. I'm Marcel, and could you explain to us why does Spain has different regulation regarding for the stem cell transplantation and for the cellular therapy, and how do actually they differ from each other? Like the definition of the stem cell transplantation, why it's different from cellular therapy. Thank you.
Thank you, Dr. Marcel. Yeah, the difference regarding the regulation are really small. So basically, the concept of minimal manipulation of cells for transplant and cell therapy is the same. Only what we have some difference between some countries is the environment that is required to process cells for starting material. For instance, in Spain is needed a class D environment, and in other countries like Germany is required to a high restricted level like class C. So this is in a regulatory point of view, but the other requirements are the same between European member countries regarding the traceability that you have to use it, the quality control, the sterility test. They are the same basically in all the European countries.
Okay, Dr. Jesus, thank you for your explanation. Very interesting presentation. Thank you very
Dr.
much.
Marcel. Thank you.
Okay, if there are any participants that want to ask Dr. Jesus again, please you can chat. You can write in chat below, or you can raise hand for ask directly. Oh, okay. There is a participant, Dr. Arif Rahmat, want to ask you directly. Please, Dr. Arif.
Thank you, Dr. Dika. Good morning, Dr. Jesus, and good afternoon, Dr. Dika. First of all, I would say congratulations for your presentation. It's very mind-blowing for us, especially for me. It's absolutely a great presentation. And since the stem cell is a very new things for us, and especially for me, I would like to ask a few questions about stem cell. Recently, I always listen about there's two different stem cell, hematopoietic stem cell and dendritic stem cell. Would you please explain me about the difference between those two things? And the second question is there any specific laboratory examination to check after the patient have the treatment from stem cell therapy? Thank you.
Okay, thank you, Arif, for your question, and yeah, really thank you for your words. Regarding the first question of dendritic cells, dendritic cells are cells that present the antigens, so they are commitment to present an antigen to T cells to provoke reaction or immunology response. So stem cells, their commitment is to renewal in order to provide more stem cells. And then the other commitment is to differentiate in hematopoietic stem cells line. That it means lymphocytes, megakaryocytes and platelets, erythroblasts, red cells, neutrophils. So this commitment of the stem cells can derivate to the dendritic cells that are presented cells that they go to the thymus to be trained, and then they are able to present the antigens to regulatory T cells, CD4 basically, that they express the HLA class 1. So this is how the body can attack infections or tumor cells. So they are really important in our cytological effect. In fact, they are now using dendritic cells in order to develop vaccines or advanced therapy. So this is a new way to treat patients. And regarding your second question, I'm not sure if I understood completely well. If you mean that what are the requirements of a stem cell laboratory setup, if that's the question, the requirements has to be defined by your authority. So what I would expect is in Indonesia, there was authority, maybe transplant authority that define a minimum requirements to set up a laboratory and to be authorized. At least this is how it works in Europe. The authorities authorize the activity of a cell processing laboratory, the procedures, the storage, the distribution, and then this authorization lasts during 4 years. Every 4 years, you have to renew this authorization, and moreover, cell processing laboratory can apply for international standards. In some countries, they are mandatory, but in others are not mandatory. For instance, JC. But JC standards, in the last years, it was not mandatory in Spain, in contrast to other countries like Netherlands, that was mandatory that one center is a JC. Right now, transplant centers with allogenic transplant program, this is mandatory to have JC accreditation, not only the clinical team, but also the collection and stem cell processing. So this is a decision of the authority, and recently the Spain Transplant Organ Authority make a directive that it's mandatory to all the centers with unrelated and related transplant program to have JC. So the requirements is defined by the authority basically, and if you apply the standards, also there is a lot of standards that you have to meet, and this is really helpful if you want to set up a transplant program and cell therapy laboratory to meet PAC-JC standards, because it will help you to understand what are the requirements.
Okay. I think that Dr. Arif want to ask you about the laboratory test after post-transplantation for the AACT. Maybe like engraftment laboratory test. I think that Dr. Arif asked for that. What type of laboratory test that we can perform after the post-transplantation?
Thank you very much for this question. Regarding after transplantation, in our setting, we perform the typical engraftment of neutrophil and platelet that can be done by clinical laboratory, but also we test the chimerism. The chimerism is performed usually after 1 month of allogenic transplantation in order to test the percentage of cells that are from the donors than those that are from the recipients. Regarding the type of transplant, you usually expect that more than 95% of the cells are from the donors. So there are different technologies to do this test, and the most typical is NGS technology and tandem repeat. So usually the immunohematology laboratory are the responsible to do this kind of test. And yeah, this is a really important test in order to anticipate, because for instance, in cord blood transplantation, it's really important to test the chimerism. If you don't have a really high chimerism at the 3 or 4 weeks after transplant, then the risk of graft rejection is really high. So you have to prepare just in case it happens. And also, you can manage the relapse. If you have a patient with full chimerism, and in the next control, you see that the chimerism is losing, then you have to do something. Maybe you have to remove the immunosuppressive, or you have to provide a donor lymphocyte infusion in order to revert the chimerism. So this is a tool that is really interesting if you have allogenic transplant program.
Thank you, Dr. Jesus, for your answer. Is there more questions from Dr. Arif?
No, thank you. That's very good answer and explain very much. Thank you, Dr. Dika.
Thank you, Dr. Arif. Is there any participant want to ask Dr. Jesus again or directly? You can raise your hand. Okay, I think that's all. And thank you, Dr. Jesus, for sharing your experience and knowledge. We are very grateful to have you here today. And we hope that we can continue our discussion in the other time. Okay. Dear participants, we have reached the end of our discussion today. I hope the knowledge from Dr. Jesus can help us of giving a better care for the patient. And thank you, and I will hand it over to Dr. Mitra. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you very much, for the invitation, and I hope we can see in the future. I can visit your center. Thank you very much for your kind invitation.
You're welcome.
Thank you, Dr. Dika and Jesus Fernandez Sojo, MD PhD, for a very interesting lecture and discussion. Finally, we arrived at the end of this guest lecture. Thank you very much for our speaker, Jesus Fernandez Sojo, MD PhD, our moderator, Dr. Maherdika Sania Maherdika, clinical pathologist, our respected professors, attending doctors, residents, and all the audience. We would like to remind all the participants to fill in the attendance link, which can be accessed in the meeting chat. In this guest lecture, you are going to get an e-certificate with the Pelataran Sehat SKP. To the honorable professors, doctors, and all participants, before leaving the Zoom, we are pleased to take a picture together. To Jesus Fernandez Sojo, MD PhD, Dr. Moderator Sania Maherdika, clinical pathologist, Dr. Dwi Ratningrum, clinical pathologist, consultant of infectious diseases, Dr. Nyoman Suci Widyasiti, Magister of Health, clinical pathologist, consultant of hematology and immunology PhD, Professor Banundari Rahmawati, clinical pathologist, consultant of endocrinology and metabolism, consultant of blood bank and transfusion medicine PhD, and Professor Dr. Lisyani Besuromo, clinical pathologist, consultant of nephrology and respiratory, consultant of clinical immunology. Please kindly open the camera because we are going to take a photo together. On the count of 3. 1, 2, 3. Once again. 1, 2, 3. To all participants, please kindly open the camera on the count of 3. 1, 2, 3. Next. 1, 2, 3. Once again. 1, 2, 3. Thank you. Ladies and gentlemen, this ends the guest lecture today. We would like to thank Jesus Fernandez Soho, MD, Ph.D., as the guest speaker. Furthermore, to all participants, thank you for joining us today. Hopefully, all the knowledge that has been given can enrich inside for all of us. To all committee and participants, thank you. We do really hope to be able to see you again in the next occasion. On behalf of our committee, I would like to apologize for the inconveniences and mistakes during the event. Stay happy and healthy. Assalamualaikum warahmatullahi wabarakatuh.
Okay, thank you very much, Professor Fernandez and Dr. Maherdika, and for your all attendance today. Thank you very much.
Thank you, Dr. Jesus.
Thank you very
Thank
much.
you,
very much.
Summa Pandita Doar Maherdika. Thank you. Thank you, Dr. Suci. Thank you,
Thank you.
Dr. Suci. Very good. Prof. Lis, sehat ya?
Halo, Dr. Sunarto.
Sehat, sudah sehat. Umur panjang,
Iya.
sukses selalu. Yang penting
Makasih.
sehat. Prof. Banundari sehat. Saya izin live ya. Hey, kawan lama. Izin live. Saya mau izin live. Terima kasih.
Makasih. Ini ada link absensi enggak ya?
Sudah diisi, Prof. Tadi ada di bagian 1.
Ya. Belum saya lihat.
Ada di bagian 1. Yang paling atas.
Paling atas. Waduh.
Iya.
Sebentar.
Saya mau izin live ya, profesor semuanya, guru-guru besar.
Yuk.